This is the third and final video for chapter 1, and this video is going to focus on the scientific method. So we're going to look at what scientists do to study the world around us. So here's the scientific method. I know there's a lot of text on here, so I know you guys can read this yourself, so let's kind of just walk through the gist of what's going on. So scientific investigations always start with an observation. Okay, and when we see something, like for instance, if I drop a pen, all right, if I'm holding a pen and I let go, it's going to fall down. If I pick up a stapler and I let go, it's going to fall down. If I hold up a textbook and let go, it falls down. So before, right, we understood gravity. That was observation. Why is everything falling down? What is happening here? So observations lead to a question. So we always start at an observation. That leads to a question. Why does everything fall down? And then that leads to a hypothesis. Now, please highlight these features of a hypothesis. Hypothesis is a possible explanation for what you saw. Okay, so something we could say now is, I think there's gravity, and that's what's causing these, objects to fall to the ground. Okay, but it's a little more complex than that because this possible explanation has to be testable. We have to be able to do some sort of experiment and figure out if we're right. And then another feature is that it has to be falsifiable. That means it can be proven false or incorrect, or it can be proven true. Okay, so either we're right or we're wrong. Another feature of hypothesis is that it is an explanation, not a question. Notice that question and hypotheses are two different things. So this is why does it happen, and this is it happens because. Okay, so again, it's an explanation and not a question. It is testable and it is falsifiable. All right, so once we have a hypothesis, we're going to make these predictions. That is going to help us set up our experiment, which we are then going to conduct the investigation. While conducting the investigation, we will be collecting data. Data comes in two forms that I want you to know the difference between. Quantitative data is one form. What does quantitative sound like? Quantity, right? And quantity is a number. How many apples are you buying? So quantitative data comes in the form of numbers. Qualitative, what does that sound like? Sounds like quality, right? So how I remember it is that qualitative data are descriptions, because the better the descriptions, the better the qualitative data. So imagine if you're brand new to town and you're like, well, where's HEB? And somebody says, well, you're going to get onto this one road and then you're going to turn right onto this other road and you're going to see this building. That's how you know you're on the right track. Keep on going. It's down on the right. That's not very good. It's not very helpful, right? But if they say, go down University away from campus, you're going to turn right onto Texas. You're going to see the Bonfire Memorial on your right. Keep on going down. HEB is going to be right there on the right-hand side of Texas. That's a much better description. That's better qualitative data because it's of higher quality. Once you have your data, you're going to analyze that data and come to a conclusion. Was your hypothesis right? Was it not? Now from here, normally if your hypothesis was right and we've learned something here, you can take your data and you can write it up into a journal article where a lot of other scientists in the community are going to review it. They're going to point out flaws in your experiment, maybe other things that might be going on. They're basically going to critique you. Very rarely do they say, great job, accepted. I've never known that to happen to anybody in all my years as a scientist. And so they normally say, hey, we'll try this experiment, include this control, and then let's see what happens. Once the scientific community is happy, which can take quite a bit of work, you can publish your results. Okay? So when we're setting up the experiment, this is probably the hardest part because there's a lot of things you have to think about because you want to keep everything constant except one thing. Okay, so we're going to use this example of scientists that want to determine which type of coffee bean contains the most caffeine. Okay, so don't worry about how we extract caffeine from coffee beans. It's just a very simple example. So if we're trying to look at which coffee bean contains the most caffeine, we want to make sure that the only thing changing in our experiment is the type of coffee bean. We want to make sure they're all planted in the same type of soil, that they get the same amount of water, the same amount of sun, that they're the same age when we pick those coffee beans or when the caffeine is extracted. So everything needs to stay the same except the type of coffee bean. So we say that the thing that's being manipulated is our independent variable. So that's what is changing. So for instance, the type of coffee bean is what's changing. The second type of variable is the dependent variable. That's what we're measuring. So look at our example. Determine which type of coffee bean contains the most caffeine. So we're trying to measure the amount of caffeine that we get from our different types of coffee bean. So that means what's being measured here is how much caffeine comes from each type of coffee bean. So the amount of caffeine extracted is our dependent variable, and our type of coffee bean is what's the independent variable. And then everything else is a constant, or we call it our standardized variable. So in this example, I just mentioned that we're extracting caffeine from 3 grams of each type of coffee bean. So that's a constant. So we call that a standardized variable. But again, had I told you about the temperature they were grown at, the type of soil, all of those would be constants as well. Okay, so we always need to do a controlled experiment. You have to have something to compare it to. So maybe, we know how one type of coffee bean, well, actually, let's change the scenario a little bit. Now we want to know, well, what type of soil is best for growing Costa Rican coffee beans. Okay, I'm completely making this up, guys. So if you're a coffee expert, I'm sorry if I butcher this. So maybe now we're looking at what type of soil is best. Okay, and so that means I know how what type of soil is common in Costa Rica. So that is my control. I know how these Costa Rican coffee beans grow in a Costa Rican soil. So then maybe now I want to try a different type of soil. Okay, maybe I want to try 3 different types of soil. So my control is the Costa Rican soil because we know what that results in. Okay, that is our comparison. Our independent variable would now be the type of soil. Maybe I have one that's more acidic, one that's more basic, one that has more clay in it. So that would be my independent variable, the type of soil. And my dependent variable is still the same. It would still be how much caffeine can be extracted. So again, your control group is your baseline, and the experimental groups are what is being manipulated. So again, that would be the coffee beans that are being planted in the acidic soil, the coffee beans being planted in the basic soil. So I'm going to upload a few examples under the resources portion of the class that has, practice experimental designs, and you're going to get some practice identifying the independent variable, dependent variable, constants, controls, and things like that, because you definitely want to be able to look at a scenario and identify these variables and control groups and experimental groups on your exam. So to wrap up this video, let's talk about the difference between a theory versus hypothesis. So a hypothesis was one simple explanation that was to serve as a potential answer for one specific question. Well, a theory is much more broad. We're looking much bigger picture here. It's based on years and years of data that's been collected from hundreds of different experiments. So, for example, a theory is the cell theory. All living things are made up of cells. Well, that's a lot of data, right? We've looked at hundreds of different bacteria. Well, they're made up of cells. We've looked at hundreds of different dogs, cats, birds, swans. All of them are made up of cells. So that's why theory is much more broad. It's much more accepted because it's based on a lot more information. And finally, scientific inquiry or investigations have its limits because how I interpret a set of data can be different how somebody else interprets a set of data, right? You've all seen instances where, there's a picture and 2 people are looking at it and one sees a face and one sees something totally different because how people see things and interpret things are different. That's what makes us all different. Some hypotheses cannot be tested yet just due to limits with technology or access. For instance, trying to do experiments that have to do with space can be hard, right? We can't go out and round us up a star or collect a bunch of moon samples all that easily. So some hypotheses can't be tested just due to our human limitations. And some data can be viewed skeptically for non-scientific reasons. People don't want to believe in global warming, so they choose not to. Doesn't matter what the scientists who have all these advanced degrees and are experts in their field, doesn't matter what they say. If people don't want to believe something, unfortunately, they won't. So there are limits to science and scientific inquiry. That wraps up chapter 1.